Good morning, everyone. Thank you for attending today's webinar on quality control. So there are mainly four different stages in which NGS data is analyzed in the tool. First, we import aligned samples, which are either in SAM-BAM format or Illumina-specific aligned formats or generic bed format or compressed versions of these files. We support multiple vendor platforms, Illumina 454, ABI, IonTorrent, and PacBio. We also support the different library layouts, including single-end, paired-end, and mate-paired. So once you have these aligned files and you import them into the program, the next step to do is to carry out quality control. There are different types of quality control: generic quality control, vendor-specific quality control, and application-specific quality control. And after performing the necessary quality control, we then perform the analysis, the application-specific analysis. This could be RNA-seq, DNA-seq, ChIP-seq, or even small RNA analysis, which will be coming out in a couple of months. Once the results are generated by these analysis steps, we can verify the results in the genome browser or custom visualizations. We can perform downstream analysis on the gene list, such as Go enrichment, GACA, NLP, and pathway analysis. Today's agenda is about quality control. As I mentioned earlier, we can think of quality control in this dimension, where you have generic quality control, where we examine the bases and their qualities. We have vendor-specific quality control, where we look at the quality summaries along instrument-specific units like lanes and tiles. And then we have experiment-specific QC, that is, the quality control that should be carried out before running any specific analysis, like ChIP-seq might have its own quality control, DNA-seq, where you're discovering mutations, might have its own quality control, etc. So let me take you through these step by step. Overall, the quality control elements are present at the top of the workflow, which is always shown on the right-hand side of the tool. There are two sections to quality control. One is quality inspection, where we check the qualities of the bases and get an idea of the distributions of the average base qualities, average alignment scores, etc. And the second step is filtering, where based on the information that has been shown in the quality inspection, we decide the thresholds and filter out the bad data. There are many NGS filtering tools available on the command line, but one problem is you do not know what thresholds to use in filtering the data. If the average base quality of all the reads is only 20, but if the visualization does not show that information, then it's hard to know what thresholds to set in filtering out the reads. So that is why in Avadis NGS, the quality control is a two-step process. We first inspect the qualities, decide upon the thresholds, and use those thresholds in the filtering stage. So first, let's talk about generic QC. All NGS instruments generate reads. A read is a sequence of bases like A, C, C, G, A, and associated with each base, there is a Phred quality, which indicates the confidence that the sequencing instrument has on that particular base. So the base quality is actually a Phred score, and it is defined as -10 times log 10 to log base 10, the probability of an error. That is, P is the probability that the base is being called incorrectly, and we take that probability, take a log of it, and multiply it by -10 to obtain the Phred quality score. So what do these Phred quality scores look like? If the probability that the base is wrong is only 0.001, that is 10 to the power of -3, then the Phred quality corresponding to the base is 30. Similarly, if the probability of a wrong base is 0.01, that is 1 in 100, then the Phred quality that corresponds to it is a 20, etc. So if the average Phred quality of a read is 10, it means that at an individual base location, the probability that the base is being called incorrectly is 0.1. So if you have a 50 length read in which all of the qualities are 10, then it is possible that 5 out of those are sequencing errors. That is one way of interpreting the base qualities. So the first thing to do is to check did we have a good sequencing run or not? That is, how many of the bases have quality 2, how many of the bases have quality 3, what is the maximum quality that we have, etc. So this plot shows that the majority of bases that were obtained in a particular sequencing run had Phred quality score 34, which is quite good. Here is a second sequencing run. You can see that a lot of the bases have excellent quality of 50, but you also have a few bases which are quality in the single digits. Instead of looking at individual bases, we can also look at the average of the qualities summarized along a read. That is, we take a read, we take the qualities of all of the bases in the particular read, we take the average, and we call that the average base quality of a read, and we can look at the distribution of the average base qualities. So here it shows that there are approximately 30 million reads which have an average quality of 33. We have around 3 million reads with an average quality of 32, etc. So overall we have a very good base quality distribution of the reads. By the way, some of these visualizations are not present in Avadis NGS version 1.2.3, but they will be present in the upcoming version 1.3, which is due to come out in a couple of months. And the slides in which the novel visualizations are being shown are marked in the bottom right hand corner with an NV. So if you were to use the filtering steps and filter out reads with poor base qualities, then we might see a truncated distribution, or we could also see a truncated distribution because the base qualities in the file are not really Phred base qualities. This often happens when you import Illumina files. Illumina uses a slightly different standard for representing the base qualities, because of which you might see an offset. That is, you might see the quality starting from 33 and going on towards 63. What we should do is subtract 33 from these values and shift them to the 0 to 33 scale. So based on these quality distributions that have been shown, one can then move on to the filter step, where we can specify that all reads with an average base quality below a certain threshold need to be removed before further analysis. Another problem one often has in NGS analysis is that portions of the adapter can remain in the read and cause problems for downstream analysis. So there are two ways of identifying these adapter sequences. One is to take a sample of the reads, look at the first 20 positions and the last 20 positions of the reads, align those 20 positions across all of the reads or a large sample of the reads, and see if any particular motif stands out. Here, for instance, if we take on a particular sample, if we had taken 100,000 reads and computed the logo of the first 20 bases, ideally we should see a uniform distribution of ACGTs, but what we can see out here is that the first base of all of the reads is a T, which is why we see a strong T signal out there. We can also see that the second base is predominantly an A, because the A is slightly larger than the other bases, and similarly the third position is also slightly more disposed towards an A. So there seems to be a TAA signal at the start of the reads, and after that you can see a uniform distribution. This is an indication that an adapter sequence of length 3 might have been left at the beginning of the reads. Whereas towards the end of the reads we see no such signal, which means there is no 3' adapter here. These are indications that the reads need to be trimmed before alignment. Instead of just looking at the logos which give out only strong signals, one can also look along the entire length of the read and see what is the percentage of ACGTs at each particular read position. Again, you would expect the distribution to reflect the GC percentage of the genome that is being sequenced. But if you see any variations out here, for instance, here we can see that after the 55th cycle, the percentage of Ts has taken a dip. That could be an indication that there is something going wrong in the sequencing. Maybe the reagent amounts are getting low, etc. Again, this visualization will appear in the next version of the tool. The nature of most sequencing is that the quality of the bases in the initial flow cycles is very good and the quality dips towards the end of the sequencing cycle, and that is reflected by a quality plot where for every position along a read we plot the average quality of all of the bases in that particular flow cycle. So you can see that the very first base has excellent quality around 67, and as we go towards the end of the read, as we approach the last portion of the read 79, we can see that the median quality has degraded to 57. We now move on to vendor specific QC steps. In Avadis NGS we have QC plots specific to the Illumina platform. The Illumina flow cell is shown in this particular slide. It has 8 distinct lanes. Each lane has multiple imaging units called tiles. The number of tiles can range anywhere from 100 to 120 or even a little bit larger. Each tile has multiple clusters, could be as many as 30,000 clusters, and the cluster will ultimately result in a read. Reads arising out of the Illumina platform encode the lane and tile information in the read IDs, and using these attributes one can perform Illumina specific QC steps. To be more specific, an Illumina read ID is shown in the picture above, and you can see that it encodes the run, the lane, the tile, the XY position of the cluster within the tile, the mate number for paired end sequencing, and an index for multiplex samples. In addition, the read also has the read sequence, which are the base calls, and the base qualities. So there are multiple dimensions along which we can summarize information and do quality control. 2 dimensions that we have decided to summarize information along are the lane and tile dimensions. So this is a tile-based quality plot. It shows for a particular sample and a particular lane. It shows for each tile of the lane what was the quality of the first flow across all the clusters in the tile, what was the quality of the second flow across all the clusters in the tile, etc. What we would expect is that the initial flow cycles are of good quality, so we would expect high qualities towards the left hand side of the plot, and as the flow cycles keep progressing, you would expect a deterioration of quality and more of a blue color towards the right hand side of the plot. This helps to spot if there are any aberrant tiles, in which case you would see a sudden shift to blue where you would not expect it. For instance, in this particular picture, we can see that in the very first tile, the very first flow is shown in blue, which means the quality was poor. Whereas for tile number 2 and other tiles, the color is in red, which means the quality is quite good. In general, type of quality plot that you should see is out here. You see a mainly red picture on the left hand side and a deterioration towards blue on the right hand side. But depending on the order of the tiles in the lane, you see different types of pictures. Here you can see a symmetric picture where the first half of the tiles are a mirror image of the second half of the tiles. That depends on the order of the tiles in the lane. The picture that we see out here is also similar, and occasionally you will spot a bad tile. For instance, out here we can see that tile number 112 has a poor quality because it starts fading to blue very early on, and in this picture we can see that the first set of tiles produce reads of good quality, but the second set of tiles, the quality is a little bit poorer towards the latter half of the reads. One can then, using the filter, one can isolate the reads which originated from a particular tile. Then once we create a separate read list which has only the reads which came from a particular set of tiles, we can open the read list in the genome browser to see what are the mismatches present in those particular reads. The mismatches are indicated by dots in the reads itself and also by the mismatch histograms in the coverage plot. Another way of checking the overall alignment quality of these reads is to use the tile alignment quality plot. So the tile alignment quality plot packages a lot of information, so let me take you through that in detail. On the x-axis we have all the tiles of lane 7. And what we want to do is take all the reads which originated from a particular tile and see what was the fate of these reads during the alignment process. The fate could either be that it did not match anywhere at all, that is indicated by NM in the legend in the bottom right hand corner, or the read was not processed because of quality control issues, QC. The R0, R1 and R2 indicate that when alignment was performed on a particular read, it aligned in multiple locations. R0 means it aligned in multiple locations, but there was no mismatch. R1 means it aligned in multiple locations with a single mismatch. R2 means it aligned in multiple locations with two or more mismatches. Similarly, U0, U1 and U2 mean that the read aligned uniquely with 0, 1 or 2 or greater mismatches respectively. So with that legend in mind, let's take a second look at the plot. If you focus on any particular tile which has approximately say 38,000 reads, then we want to know how many of those reads aligned uniquely, how many of those reads did not match at all, how many of those reads aligned in multiple places, etc. And that is what is shown by the scatter plot. Okay. So ideally what we would want to see is a pink line which is much higher than any of other colored spots, and that indicates that the majority of the reads originated from that tile aligned uniquely without any mismatches. The tile alignment quality shown out here is an example of an almost perfect sequencing run in which none of the tiles went bad. Sometimes you do see tile alignment qualities as shown out here. If you look towards the right hand side, you will see that close to tile 112, you will notice a red spot that indicates that a majority of the reads which originated from the tile did not match anywhere at all on the genome. And that is because, if you remember the base quality plot that I showed a couple of slides earlier, that is because that particular tile, the quality of the bases went bad very early in the flow cycle. So that concludes the vendor specific QC. We'll now move on to experiment specific QC and concepts like alignment score and mapping quality. So alignment score is a term that we have introduced in the tool to summarize the number of mismatches, insertions and deletions that are present in a read. For instance, let us take an alignment. In other words, the matching of a read to the reference sequence. Here we have shown the reference sequence in the second line and a read sequence in the third line, and the alignment is a classic DNA sequence alignment like those produced by BLAST, where we use dashes to indicate insertions and deletions. One can see that in this particular alignment there are two single nucleotide mutations at the 4th location and the 7th location. There is a 2 base pair insertion, that is, there are 2 bases A and C that are not present in the reference but that are present in the read, and there is a single base pair deletion. So one way of quantifying the amount of difference between the reference sequence and the read sequence is to take all the locations, count the number of locations at which the reference and the read match exactly, and divide by the length of this alignment. So here the length of the alignment is 20, and we have 15 locations where they match exactly. So the alignment score of this particular alignment would be 75. During the sample import process, Avardis NGS calculates the alignment score of all the reads and displays the distribution of the alignment score in the quality control process. However, because the gapped alignment between the reference and read is not present in the SAM or BAM files, because it would make the files extremely voluminous, it relies on a CIGAR. A CIGAR is a summary of the differences between the read and the reference. So the CIGAR for the alignment shown in this particular picture would indicate the locations of the mismatches and the insertions and deletions. For example, we could read out this CIGAR as the alignment comprises of 3 matches, followed by 1 mismatch, followed by 2 matches, 1 mismatch, 3 matches, 2 insertions, 3 matches, 1 deletion, and 4 matches. So the cigar fully specifies the pattern of matches, mismatches, insertions and deletions, though it does not tell you which of the bases, what was the nature of the mismatches. If the cigar was present in this full format in the SAM file, then it provides the complete information necessary to calculate the alignment score accurately. Fortunately, in the original SAM format, which is commonly used by most aligners, the mismatches are not indicated in the cigar. Only the locations of the insertions and deletions are indicated, which means the cigar would actually look like 10M, 2I, 3M, 1D and 4M. So this cigar does not really give us the full information necessary to compute the alignment score accurately. So let's take a look at how alignment score distributions look like. You typically see a strong peak at 100, somewhere between 85 and 100 you'd see a strong, nice distribution, and then you'll see a lot of unaligned reads which are really poor quality and alignment score of zeros. Now coming back to the alignment score computation, as I mentioned earlier, since SAM files have only a partial cigar and not the full cigar, there is an additional attribute in the SAM file called the MD tag, which gives the locations of the mismatches. So by taking the partial cigar and the optional MD tag, we could recompute the full cigar and based on that compute an accurate alignment score. So if the MD tag is present in the SAM file, then during the import process we make use of it, we recompute the full cigar and use that to compute the alignment score, and the alignment score shown in the QC plot is accurate. However, if the MD tag is absent, then during the import process we cannot compute the alignment score, so the cigar is taken as it is. The alignment score distribution that is shown is actually an overestimate of the true alignment score. However, when we look at the reads in the genome browser, at that point we take the reference sequence and the cigar, dynamically recompute the accurate cigar and show the correct alignment score in the genome browser. So you need to be aware of the fact that the genome browser shows correct alignment scores, but the alignment score shown in the distribution might be an overestimate. So here are some more alignment score distributions, and here is a look at the genome browser. As you can see in the genome browser, since we recompute all of the cigars and the alignment scores, you can see that in the spreadsheet at the bottom, you can see the accurate, the full cigars with the lowercase m's indicating mismatches, the uppercase m's indicating match characters, and the recomputed alignment scores. In a month or two from now, we'll be releasing Avardis NGS 1.3 with support for small RNA analysis. In small RNA experiments, the read length is anywhere between 15 to 30 base pairs. Because of the small read lengths, alignment scores make it hard to estimate the number of mismatches. So instead of showing the alignment score distribution, we will be showing the mismatch count distribution in small RNA experiments. So you can see that in a sample that we have processed recently in-house, around 2.6 million reads have 0 mismatches, around 600,000 reads have 1 mismatch, and you have around maybe 250,000 reads with 2 mismatches, etc. Next we move on to the concept of mapping quality. So suppose we have the reference chromosome shown in blue, chromosome 1, and we have a read which has been aligned against it by the aligner. The mapping quality is an indication of the confidence that the aligner has that the read really originated at this particular position. So what do we mean by that? Suppose there are 10 other identical genomic locations present in the genome, then the read could have originated from any one of those other locations also. So the probability that the read originated in a different location is 9 over 10. We can take that probability and again calculate the Phred score, and that is the mapping quality. Okay. So in general, the mapping quality is an indication of whether the read is present in a repetitive location or a unique location. But But it's actually a little bit more complicated than that. Suppose the read does not have any other matches in the genome. This is the only place in the genome that it matches without any mismatches. It still doesn't guarantee that the mapping quality will be perfect. For instance, suppose we have another location in the genome, say on chromosome 10, where the read could have matched with one mismatch. And the probability that the read actually originated on chromosome 10, and because of a sequencing error at that precise location, it got mapped to chromosome 1, is proportional to the base quality of that location where the sequencing error was supposed to happen. So in other words, it is possible the read originated on chromosome 10 and that it had a single sequencing error, and because of the sequencing error, it aligned perfectly to chromosome 1 instead of to chromosome 10. So all of these need to be taken into consideration by an aligner which outputs accurate alignment mapping qualities. So the mapping quality, to summarize, is a complex function of alignment score, the mismatch location, the base qualities at the locations of the mismatches, the repetitive content of the genome, and it becomes even more complicated when you're aligning paired end and made pair libraries. So let's take a look at some of the mapping quality distributions that we have seen in our samples. In the SAM file, the mapping quality was originally conceived as a number between 0 and 255. 255 is supposed to be used as an indicator that the aligner does not know how to compute the mapping quality. So the values that are actually supposed to be used are between 0 and 254. So one mapping quality plot that we have seen is as follows. Here the maximum mapping quality is 254, and if a read aligns uniquely, it is probably assigned 254. If it aligns in two places, then the mapping quality is halved, so you get 127. If it aligns in three places, it's divided by a third, so it becomes 80, and so on. So this is pretty much a reflection of the number of matched locations. So one must understand that this is not a true Phred quality mapping quality, but it is the aligner's own interpretation of mapping quality. Most aligners are not capable of computing mapping qualities according to the definition, and they put in their own versions of the mapping quality. Unless the end user looks at the mapping quality distribution, they will not be able to take a call on whether it's a Phred-like mapping quality or it is some other aligner's interpretation of the mapping quality definition. Here is the mapping quality going anywhere from 0 to 60, which is a more realistic Phred scale, and I think this mapping quality was generated by BWA or MAQ. And finally, here is a sample that came out of the Illumina ELAND pipeline. And you can see that the mapping quality scale is completely different. That is because ELAND's mapping quality definition is different from the Phred quality distribution definition that I mentioned earlier. So the mapping quality can have a large effect on the results of SNP analysis. In the SNP analysis workflow, there is a checkbox indicating whether the SNP analysis should use the mapping quality or not. By taking a look at the mapping quality distributions, the user needs to take a call on whether the mapping quality looks like a Phred mapping quality or not. If it looks like a Phred mapping quality, then that should be passed as an input to SNP calling, and the SNP calling results will be more accurate. So here one can see in the genome browser that there are many reads with 100 alignment score, but the mapping qualities are poor, and that is probably because the read is in a repetitive region. Next, we move on to read pair status. This is useful for paired end and made pair libraries. In paired end and made pair libraries, the unit that is sequenced is two reads which are expected to have a certain orientation and distance separation from each other after alignment. Based on the expected orientations and distance, a status is assigned to each read pair. The normal status indicates that the two reads which form the read pair matched at the expected distance and orientation relative to each other. If one of the reads were flipped, then it would cause then the status assigned to the read pair would be one made flip. If both of the orientations are unexpected, then it would be assigned a status of both made flip. And similarly, if the two reads have the proper orientation relative to each other, but they align much closer than expected, then the status would be made near. If they align much further than expected, the status would be made far, etc. In general, the number of normal status read pairs plus the made far and made near status read pairs should occupy around 80% of this pie chart. That is what we have seen in most of the normal samples that we have analyzed in-house. If you see a large deviation from that number, it is possible that the alignment is incorrect. If you have a large number of chromosomal aberrations, then reads which are expected to align a certain distance from each other may align much further or much nearer depending on whether there are insertions or deletions type structural variations, and those might reflect in different pie charts. However, in some of the samples that we have seen so far, in the normal and the tumor, it is very hard to differentiate purely from the pie chart whether there are a large number of structural variations or not. Next, we move on to targeted regions QC. For researchers who are sequencing exome samples, one way to verify whether the sequencing is correct is to visually inspect the coverage plots in the genome browser and check whether the coverage peaks correspond to exon locations or not. A second way to do it would be to create a region list of all the targeted regions, and then using a script that is available on our website, one can count the number of reads that are falling in each one of those regions. The resulting region list, which has for every targeted region the number of reads that are falling within it, that region list can be inspected in the tool using region list operations, and one can see how many targeted regions have 0 reads, how many of them have 1 read, etc. To make this process easier, we are going to have a targeted regions QC workflow link, which will compute the number of reads which are falling within the targeted regions, the number of reads falling within the vicinity or delta padding of the targeted regions, the average coverage of the targeted regions, etc. And it will do it for each of the samples present in the experiment. Another thing that we would want to verify is the insert length distribution. In paired end library preparation, during the size selection, typically one selects for fragments of say 200 base pair to 500 base pair length, and once the reads are sequenced and aligned against the genome, one would expect them to align at the same distance away from each other as the size selection protocol step. In this particular case, a sample where the size selection of 200 was used was sequenced, and one can see that even after the reads were aligned, the two reads aligned approximately 200 base pairs away from each other. So there's a good sequencing run. If the median insert size computed from the alignment statistics was very different from the size used during library preparation, that could again indicate quality control issues. Finally, we move on to application-specific QC for ChIP-seq. In ChIP-seq analysis, most of the peak finding algorithms assume that duplicate reads have been removed. Here we have an example of 2 different samples which have different coverage patterns. One can see that the coverage pattern at the top is very spiky, and the coverage pattern at the bottom is a little bit more uniform, and the coverages are very high because the average coverage is somewhere around 80. If we take the sample at the top and we inspect any of those high coverage regions, we can see a nice positive and negative coverage peak. You can see that they do not seem to have any duplicate reads at all. But if we inspect a high coverage region in the second sample, you can see that there are a lot of duplicate reads out here. The presence of these duplicate reads might cause false positives in the peak finding algorithm. So one can use the remove duplicate reads filter to eliminate these reads, and after that carry out the peak finding using either PICS or MAX or the enriched region detection workflow links in the ChIP-seq experiment. Coming to RNA-seq, we do not have any sequence specific QC plots. What we do have is QC on replicate samples, where you can carry out a PCA on the samples and color them by the group or parameter and verify that the replicate samples cluster next to each other in the PCA plot. So this brings us to an end of the QC webinar.